Hey everyone, my name's Kim, and my group decided to do our presentation on ALS. ALS stands for amyotrophic lateral sclerosis. ALS is a neurodegenerative disease that disrupts the upper and lower motor neurons in the brain and spinal cord. The loss of motor neurons in the central nervous system leads to the inability of a person to control their limbs, can lead to total paralysis, and ultimately will lead to death. The CDC says there's about 17,000 people diagnosed with ALS living in the United States today. Currently, there is no cure for this deadly disease. ALS etiology. The exact origin is unknown according to the CDC because the cause differs from patient to patient. There is, however, two common types of ALS. The most common is sporadic ALS, which, as you would suspect, is due to randomness. And then about 10% of cases reported are familial ALS. Two genetic mutations that I'll discuss from the research has to do or is related to familial ALS. The last three bullets can be things that arise from either type of ALS, but are also factors that can cause ALS, since the exact origin is unknown. So immune cells can launch an attack on self. This can be seen when immune cells attack our nerve cells in ALS. Also, chemical imbalances can change chemical environments. So a prime example is that a chemical change can lead to the overproduction or underproduction of molecules or proteins that can really affect different pathways and cascades. An example is glutamate, which is an essential protein in our body, but when there's high quantities of glutamate, it becomes toxic to nerve cells, which can then cause further damage, which, as we know with ALS, it's a neurodegenerative disease, so that can definitely attribute to ALS. The last one, which will be something that I discuss with both data points, there can be an accumulation of abnormal proteins or proteins that are not processed correctly. The innate immune response is naturally present at birth. It's non-specific and remains unchanged throughout our life. Dr. Jewell has taught us some key players that are always circulating and helping maintain our immune response correctly. So we've got neutrophils and monocytes, and then the one I mentioned are macrophages, which eradicate diseased or damaged cells, secrete pro-inflammatory cytokines, and participate in antigen presentation after it consumes a foreign body and then presents the epitope. Inflammation occurs so that there can be a recruitment of other immune cells and proteins like complement to come to a specific area or the area of infection or issue. The inflammation, as we see with redness and swelling, helps so then we can get a flood of cells and blood running to that area. So the recruitment comes via chemical signals, and I just wanted to mention a few of those chemical signals. Tumor necrosis factor, interleukin, and interferon are all chemical signals used during these immune response cascades or pathways. The first data set that I'll be talking about deals with interferon beta. I just wanted to mention that in ALS, inflammation can occur in the central nervous system, and the macrophages that are residents in the central nervous system are the microglia cells. What's all the fuss about ALS? So type 1 interferons are principal drivers of gene expression changes in response to double-stranded RNA, which is an indication of something viral being present. The FUS gene normally encodes for nuclear binding proteins for DNA and RNA. It also plays a vital role in RNA metabolism, and the FUS gene even helps with production of a variation of certain proteins. So it helps code for a protein that has different variations that it can become. So the FUS gene helps determine what proteins are created. So it is a pretty important gene. Interferon beta is involved in the innate immune response because it's known for its antiviral activity. It helps warn surrounding cells to prepare for a possible infection, or warns them that there is an infection present close by. It also helps balance immune response. It also helps balance the immune responses that promote antigen presentation or the functions of natural killer T cells. Interferon beta is also known to help dampen the inflammation pathway and the cytokine production. Familial ALS patients suffering from one of 80+ mutations in the FUS gene can have a cytoplasmic overabundance of FUS, or an aggregation of FUS proteins or FUS mRNA inside nerve cells. So the first study we're going to look at that deals with the innate response was looking to see, is FUS mRNA upregulated downstream or upstream from interferon signaling? If you look to the left at the graph that's being depicted, is mRNA levels in wild-type cells measured in the presence of interferon beta after 0, 4, 8, and 24 hours. So the y-axis is showing the relative levels of mRNA from FUS mRNA, and the x-axis was different increments of time that the samples were looked at. So 0 meaning no interferon beta present, and then 4 hours after interferon beta was present, 8 hours, and then 24 hours. When comparing the 4-hour mark to the 0-hour mark, there's a significant increase in the FUS mRNA levels that are present after the treatment of interferon beta. There were lower levels of mRNAs after 8 hours when you're looking at it compared to 4 hours, but the comparisons are actually to each 4, 8, and 24 are being compared to the 0 hour. So there was still significantly higher amounts of FUS mRNA present at the after 8 hours than there were present after 0 hours when no interferon beta was present. mRNA levels continued to decrease to the 24-hour mark, but when comparing the 24-hour mark of having interferon beta present after 24 hours being compared to when no interferon beta was present, there still was significantly higher levels of mRNA present when interferon beta was there. So the study concluded that interferon beta alone causes the upregulation of FUS in wild-type cells. The adaptive immune response. As we learned, the adaptive immunity needs to be acquired from exposure. It's very specific, and it also builds memory. White blood cells, like memory B cells, are created for long-lasting immunity, and B lymphocytes can also become plasma B cells, which produce antibodies. This helps us when there becomes an invasion and that we already have antibodies for. Plasma cells can immediately start producing more antibodies, so we can fight the infection or foreigner faster. So T lymphocytes can be a number of different types of cells, one being the CD8 T cell, which I'll talk more about, and that's a killer T cell. CD4 T cell, which is a helper T cell. Tregs, as you would assume, are regulatory T cells that can help dampen the immune response if it's too excessive or if it's time to slow down because the infection has been cleared. And T lymphocytes can also become memory T cells, which just helps us respond even faster when a foreigner comes back. In ALS, mutant CD8 T cells can induce motor neuron death, which aids in the progression and onset of the disease. So pretty much what can happen is our good guys can go bad. So the CD8 T cells, even though they're called killer cells, they're helpful because they kill things that we don't need, that are damaged or non-functional. But they can also end up hurting us and attacking our own cells, which you'll see in ALS. So as I mentioned, good guys or bad guys. Cytotoxic CD8 T cells are known as killer T cells, and they secrete perforin, which pokes holes in the cell membrane, and then it also produces granzymes, which induce apoptosis in infected cells. So SOD1 mutant CD8 T cells are CD8 T cells that express the SOD1 mutation. So these CD8 T cells were retrieved from symptomatic ALS mouse models that had the SOD1 mutant gene. The SOD1 gene codes for the superoxide dismutase enzyme, which is really helpful because it breaks down oxide radicals that are naturally formed as byproducts of normal cell function. So if we aren't able to break down these radicals, they can cause a lot of damage if they're left unattended. So this enzyme is really important. So a mutation in the SOD1 gene means that the enzyme is non-functional or doesn't correctly function. So SOD1 G93A is the exact mutation that we're going to be referring to. But these SOD1 motor neurons come from symptomatic ALS mice that have the gene mutation. It'll be easier to visualize once you see the data. So one of the first questions they asked are, does SOD1 G93A CD8 mutant T cells target motor neurons? When you look to the left at the graph, you see the y-axis is the percent of surviving motor neurons, and the x-axis is CD8 T cells, whether they're present, which is the mutated one, or none, meaning none were present at all. So this graph depicts SOD1 mutant CD8 T cells co-cultured with wild type and mutant motor neurons after 72 hours. There was no significant difference, that's what the NS stands for, when the data from the wild type motor neurons, which is the blue bars, with and without the co-culture of SOD1 mutant CD8 T cells, was compared to the mutant motor neurons, the red bars, with and without the co-culture of mutant CD8 T cells. So pretty much what it's saying is, regardless of whether or not mutant CD8 T cells are present, when you're comparing wild type motor neurons, which are normal functioning, and SOD1 mutant motor neurons, there's really no significant difference in those data sets, comparing the blue to the red. So when we're just looking at wild type motor neurons, those are the blue bars. Those are cultured from normal mice. So if you're looking left to right, the first blue bar on the left is showing 100% of the wild type motor neurons persisting after 72 hours when no CD8 T cells were present at all. The second blue bar, which is the lower one, there's a significant decrease to 60% of remaining surviving motor neurons in the presence of mutant CD8 T cells. So when mutant SOD1 CD8 T cells are present, after 72 hours, only 60% of those normal motor neurons were able to survive. SOD1 motor neurons would be the red bars, and those are taken from mice with the SOD1 gene mutation, kind of like how I had mentioned before. They're taken from these mouse models that have the gene mutation. So these CD8 T cells are expressing that SOD1 mutation. I'm sorry, SOD1 mutation. So looking from left to right again, the first red bar, 100% of the mutant motor neurons persisted after 72 hours when no CD8 T cells were present. If you look at the shorter red bar, there was a significant decrease, a little over 50%, in SOD1 motor neurons in the presence of CD8 T cells expressing the mutation. So overall, the data concluded that SOD1 CD8 mutant T cells selectively kill motor neurons, regardless of whether or not the motor neuron is coming from a mutant mouse or a normal mouse. No matter what, the mutant CD8 T cells selectively target motor neurons. So the other question that they set out to answer in this study is, do mutant CD8 T cells induce cell death in motor neurons via the MHC1 receptor? If you look at the figure on left, it's depicting the percent of surviving motor neurons expressing or lacking the MHC1 receptor after 72 hours with and without the co-culture of SOD1 mutant CD8 T cells. Again, the y-axis is depicting the percent of surviving motor neurons, and the x-axis is none, meaning no CD8 T cells, and then SOD1 mutant CD8 T cells. So the motor neurons expressing their MHC1 receptor are the blue bars. This means that they're normal motor neurons, nothing wrong with them. They have their MHC1 receptor. So when you're comparing the two of those, there is a significant difference. Less than 80% of motor neurons co-cultured with the mutant CD8 T cells survived when you compare them to the 100% of motor neurons that survived when there were no CD8 T cells present after 72 hours. Now, if you're just looking at the red bars, motor neurons treated with anti-MHC1 H2DB antibodies lacked the MHC1 receptor. So before this experiment was done with the CD8 T cells, some motor neurons were treated with this anti-MHC1 antibody, which knocked out their MHC1 receptor. So all of them, motor neurons lacking the MHC1 receptor, co-cultured with and without SOD CD8 T cells, survived after 72 hours, and you can see that looking at the two red bars. So now you can see that there was a significant difference between the normal motor neurons co-cultured with the mutant SOD1 CD8 T cells, that's the shorter blue bar, being compared to both the motor neurons that had been treated with the antibody co-cultured with and without CD8 T cells that are mutated. So when you're taking the two red bars and comparing it to the shorter blue bar, you're looking at motor neurons that don't have their MHC1, the red. Those are significantly higher, meaning they have a 100% survival rate. And then if you compare them to normal motor neurons that are also cultured with the CD8 T cells, you can see that there are less surviving motor neurons. So the study was able to conclude that SOD1 G93A CD8 mutant T cells activate cell death via the MHC1 receptor that's located on motor neurons. So in conclusion, just to revisit the results of the data, interferon beta is the chief antiviral signal in an antiviral response. So that's why the first study chose to look at interferon beta when looking at the innate immune response. So they found that interferon beta alone causes the upregulation of FUS in wild type cells. So interferon beta increases the accumulation of FUS mRNAs and FUS proteins, which ultimately increases cellular aggregation and the onset of ALS symptoms in our ALS mouse models. The SOD1 mutant CD8 T cells selectively kill motor neurons. So motor neuron cell death can increase in the presence of these mutant CD8 T cells, and that can also increase the onset of ALS symptoms in ALS mouse models. The SOD1 mutant CD8 T cell activates cell death in motor neurons via the MHC1 receptor that's found on those motor neurons. As Dr. Jewell always said, if cells are unable to bind or attach, then they cannot be activated or initiate activation. So when the T cell receptor is unable to bind to the MHC1 receptor on motor neurons, the T cell is not able to initiate apoptosis in that cell. ALS is a deadly disease that is characterized by the loss of motor neurons in the central nervous system, and the immune response can be altered if cells become compromised or do not function properly, either due to a foreign invasion or, as our data showed, genetic mutations. Both papers that we looked at, the observation and results concluded and agree with other studies that have found that the innate and adaptive immune response help shape the onset and progression of this fatal degenerative diseases. So researchers are trying to observe and change different roles of our immune system in order to see who the key players are in these diseases that are like ALS, so we can figure out what pathways or cascades are either changing and causing this disease or are eventually leading to these degenerative diseases. So understanding more about the roles of the immune responses will help us find a cure for ALS. And last but not least, these are my references. Thank you so much for listening to my presentation, and I hope you feel like you learned something.